For a while, we used to think that cellular structures called lamellipodia, Lamellii, podia. So a lamellum is a sheet, so these are sheet-like structures, so-called lamellipodia. They do not really fall under the category of tightly bundled actin filament. Instead, these are referred to as branching actin filaments. So write down branching actin filaments. Lamellipodia are branching actin filaments. So we'll talk about bundled actin. We'll talk then about actin filaments, which are sort of not bundled, but sort of random orientations, and they cross over each other inside the cell, forming a network. So we'll talk about these so-called networks. And then we probably will skip over stuff on the interaction between actin filaments and the inner surface of the plasma membrane, pretty much. And we're pretty much also going to skip over a lot of the in vitro polymerization kinetics of actin. But we will certainly talk about this. And I will talk briefly about treadmill. And the reason we're going to skip some of these topics here is because I'm wondering why I seem to be kind of behind schedule on my lecture notes and on my lectures, and then I discovered through the unfortunate confluence of the way the monthly calendar worked out this year, and the fact that somehow when Dr. Sherman and I were setting up the syllabus, I overlooked the fact that she gave herself 2 more lectures than me. So I'm really trying to make up for lost time here, so we're going to have to move rather quickly through some of the main topics of what would normally be a 2-lecture set of stuff. So. Fasten your seatbelts, as the movie actress famously said. It'll be a bumpy ride. Not so much. Here's a picture of a cell of tissue culture, a fibroblast, which has been seeded. It's lying flat on the surface of the petri dish. This is where the nucleus was. You can see the outline of the cell. You can see these little long stringy extensions coming out of the cytoplasm of the cell. We've treated the cell with gentle non-ionic detergents, which dissolves the plasma membrane, but these detergents don't have any effect on the folded structure of proteins. So when you dissolve the plasma membrane, all the soluble enzymes and soluble proteins in the cytoplasm are released, and what's left behind is what you see here in this scanning electron micrograph. These are the elements of the cytoskeleton that we see behind here. Much of what we see, or we'll say most, are actin filaments. The cytoplasm of the cell is filled with actin filaments, the most abundant protein inside the cell. Some of these actin filaments are bundled together in parallel to form these philopodia, as we'll be discussing. You extract the membranes of the nucleic acid, and a lot of the other stuff inside the nucleus is preserved as well. So then, as I said, we'll consider loosely bundled actin filaments versus tightly bundled actin filaments. And the difference is the so-called bundling proteins. If we use bundling proteins that have names like fibrin or fasciin, they have a small length, and therefore they govern a small distance between the actin filaments. If we use alpha-actinin, which we have talked about before in the context of skeletal muscle, you may remember alpha-actinin was enriched in the Z discs of muscle, and it governed the spacing of the actin filaments such that the myosin bundles can slip in between the actin filaments. So we'll talk first a little bit more detail about tightly bundled actin. an example of tightly bundled and actin filaments is in your intestine. Here's your gut in cross section. We're just looking at the bottom wall of the gut. Up here, we have the same thing. And here's in the lumen here where all your food that's being digested is. And then there's a thin surface of cells here that coats the wall of the intestine. And these are the epithelial cells. If you look at them in cross section, here they are. There's two of them, and there's a whole sheet of them going all the way down. And just as we have these protrusions, these finger-like protrusions into the gut of the cell, which help to move the food through the passage through the gut, when you look at the surface of the individual epithelial cells, you see what are called microvilli, and here's what the microvilli looks like. This greatly, hugely increases the surface area of the intestinal epithelium, so that it takes up all the amino acids and sugars and so forth, or in the lumen of the gut. And what we see here is tightly bundled actin filaments, which then spread out into the cytoplasmic structure. Another example of tightly bundled actin filaments are so-called filopodia. I showed you pictures of filopodia right here. These little things right here are filopodia. And here they are in a scanning electron micrograph of a fibroblast that's on the surface of a petri dish and moving on the surface of that petri dish. This cell is moving more or less to the left, okay? And you can tell that because in the so-called leading edge of this moving cell, we see little so-called filopodia, philo meaning, spike-like things, okay? Podia referring to the foot. At the tip of these filopodia are adhesion molecules, and the cell is growing not on a glass surface or a plastic surface of the petri dish, but rather it's a glass surface coated with a film of extracellular matrix proteins, which are part of the growth medium that we suspend the cells in. And these adhesion molecules at the tips of the filopodia adhere to the extracellular matrix proteins that coat the surface of the petri dish. These lamellipodia here, all along the ridge of these, we have adhesion molecules as well. And as the cell is moving, it shoots off these filopodia spikes and also the flat sheets of membrane, and that will wave around and can fall back and meld into the cell, or can fall forward and adhere to the surface of the petri dish and ahead of the direction of the movement of the cell. As the cell is moving in this part here is still anchored to the petri dish, and we can pull this stuff, we get pulled thinner and thinner, and usually this will break off and this will snap back into the cell body. Sometimes it gets left behind as a little piece of the cytoplasm attached to the petri dish. And there's contractile actin filaments at the myosin structure that are pulling the body forward to keep up with the lamellipodia and filopodia structures. And here are these contractile actin filaments here laying on. So these filaments are contracted on the rear end of the cell wall. Okay. These actin filaments are dispersed with myosin bundles, a loose sarcomere-like structures here. These actin filaments are spaced out the actinin, and these are what I'm calling loosely bundled actin here, these so-called stress fibers here. And here are tightly bundled actin filaments, and then the lamellipodia, which I used to consider as an example of tightly bundled. Now we refer to this as these are filaments which are branching actin filaments that are filling the interior of these little thin sheets of the cytoplasmic lamellipodia. Okay. So then we talked about tight bundles ever so quickly. Now we'll talk a little bit more about loose bundles. And the primary example of loosely bundled actin of the so-called actin-myosin stress fibers, and they interact with an adhesion structure called the focal adhesion, which we want to talk about here. So, again, now loosely bundled actin shows several examples of contractile, loosely bundled actin, the so-called stress fibers that I refer to here in red. Okay. And these stress fibers, they start one place and they end up at another place. And where they begin and where they end, they're attached to an adhesion structure called a focal adhesion. And we've highlighted focal adhesion structures here in green. Also in mitosis, as the two daughter cells are separating from each other, we form a so-called contractile ring, which produces the cleavage furrow between mitotic cells. And then along the apex of an intestinal epithelial cell here, just beneath the microvilli, we have contractile active myosin fibers. So here's another picture of stress fibers terminating in focal adhesion. So the stress fiber actin is highlighted, shows here in orange, and then in green we have the focal adhesion structures, which begin and terminate with contractile structures. So that a cell that's growing on a petri dish looks a little bit like an egg that we cracked into a frying pan. It flattens out. The egg yolk would correspond to the nucleus, okay? And the egg white would correspond to the cytoplasm here. And then along the bottom surface of this cell, we have these actomyosin bundles here. And they're contracting and they're pulling these two structures towards each other. If this were not, now this is a cell that's no longer moving, it's just anchored in place. And if this were inside of a three-dimensional living cell organism, we'd be pulling on the extracellular matrix. As the cell is moving, things are more dynamic, and we can end up sort of, this has been disassembled, so to speak, and we're pulling on these, and this one is also disassembling, and so forth. So these are focal adhesion structures here. Now, if you look at this, another way to draw this here in a cross section, here's the nucleus, is sort of plump, the cytoplasm of the cell up here. Here's the here's the upper plasma membrane, here's the lower plasma membrane, and there's space between the lower plasma membrane and the coated surface of the petri dish here. Fluid can come in and out of these regions here. But at regular sort of spot welded locations along the underneath the surface of the cell, we have these focal adhesion structures which firmly interact with the extracellular matrix coating of protein on the surface of this petri dish here. Now, it turns out, we discovered, that in the focal adhesion structures, these are centers for the production of growth signals for the cell. So that, I should say, we know that if you take normal cells, some normal tissue, break them up into individual cells, and plate them out in a petri dish, the cells are initially spherical after we go into tissue, and they'll settle down on the surface of the petri dish and then they flatten out. And then they begin to glide around on the surface of the petri dish. If you prevent them from flattening out and adhere into the dish, they don't grow. This has been referred to as the anchorage requirement for the growth of normal cells. It's the anchorage requirement for growth. And all normal cells exhibit this, except for some cells in your body, like your immune system cells, which are designed to circulate. Cancer, and the reason we're interested in the anchorage requirement for growth is that if you take cells from a tumor, a cancer tumor, and suspend them in growth medium, they don't have an anchorage requirement for growth. They can grow very well in agar suspension, for example, over the agar colonies where normal cells won't do that. This is an important distinction between normal cells and cancer cells. Invariably, when a cell is progressing from normalcy to cancerousness, and an important step that we always find is it no longer has a requirement to form these focal adhesion structures. And it turns out it's the formation of the adhesion structures which is at the root of this requirement. that's because in these adhesion structures, we have assembled growth factor signaling transition pathways here, including the famous SARC tyrosine kinase here. And just as we have from growth factors, here growth factor receptors, if they bind their ligand, then they become sticky to each other and they dimerize. And the dimerizing of the external domain by growth factors brings the internal tyrosine kinase domain bumping into each other. And that bumping into each other causes a conformational change where we partially activate the tyrosine kinase activity here. And then one catalytic domain will phosphorylate the other catalytic domain, we get full activation. These signals by themselves, we'll call this signal 1 from growth factor receptors, is insufficient for the growth of normal cells. Normal cells have to have signal 1, but they also have to have signal 2 that emanates from the focal adhesion structures. And focal adhesion structures in more detail look something like this. We have these adhesion molecules that as a class are called integrins. There are many genes that encode the subunit proteins for integrins. We have a bunch of genes encoding an alpha subunit and a bunch of genes encoding a beta subunit. So this is a heterodimer. So here's one integrin, there's alpha beta pair. There's a second integrin, there's alpha beta pair. Now, different cell types and different tissues, the integrin will be maybe like alpha 3, beta 7. On another tissue, it might be alpha 1, beta 3. Okay? And here we have a domain here that recognizes, for many integrins, a 3 amino acid sequence called RGD. So a famous extracellular matrix protein that fibroblasts bind to, that is called fibronectin. So all this stuff in here, this is fibronectin. And the integrins bind an RGD sequence in the fibronectin proteins. And in fact, this particular integrin, the first one discovered, expressed by fibroblasts, for historical reasons, is called the fibronectin receptor. The fibronectin receptor is a specific example of a particular integrin. As a class, these are called integrins. The fibronectin receptor is a member of a broad class of integrin adhesion molecules. And integrin adhesion molecules have very interesting properties that we'll talk briefly about. But as these integrins assemble, they will cluster. When they cluster, they recruit signal transduction molecules like SARC and tyrosine kinase, another tyrosine kinase called focal adhesion kinase, FAK. And we initiate the same signal transduction pathways from this complex as we would from, let's say, the epidermal growth factor receptor, the MAP kinase pathway, PLC kinase pathway, are all initiated from these structures here. Now we don't know exactly what's so mean. Why do we have to have this if we're already getting all those signals coming from the growth factor receptors, the EGF and PGF receptors? Is this a complicated thing? Do we need to have more of that signaling in order to grow? Or is there something special that's new here in the area of research now? Now, among the things that get recruited to these focal adhesion structures here, this is part of the focal adhesion, is a protein called formin, which, as we'll be discussing, catalyzes the initiation of the polymerization of actin filaments. And these actin filaments are spaced by alpha actinomeres, usually bundled actin filaments. Now, I don't know how this scientific illustration ever got past the authors. As you all know, myosin molecules are not like little Easter eggs that are popped into the middle of an actin filament. You know that. We know that myosin molecules form bipolar bundles that slip in between the actin filaments, of course. So this is a peculiar way to represent myosin. So these are loose, bundled actin filaments that are contractile because myosin bipolar bundles will slip in between. Now we're going to talk a little bit more about fibronectin, which is the extracellular matrix protein. And then we're going to talk a little bit more about this adhesion molecule, the fibronectin receptor, or more broadly speaking, integrins. So here's fibronectin. It's a it has a series of repeated short amino acid sequences here, okay? So-called heparin binding repeats here, collagen binding repeats, more heparin binding, and then a bunch of so-called type 3 repeat sequences here. Okay? Among the type 3 repeat sequences, these are pink boxes, we have two that are particularly interesting. These are two repeated sequences here, which this adhesion molecule integrin recognizes and binds to. So in these repeat sequences here, one of them has this so-called 3 amino acid RGD sequence that integrin binds to. And the actual protein itself is a homodimer, okay? This disulfide bonded to another exactly identical polypeptide chain. Okay, now let's take a look then at integrin. A very interesting feature of this adhesion molecule is that it can switch back and forth between an erect high affinity binding state and a curled up low affinity binding state. The part where integrin, and here's more detail on the curled over low affinity state here. And the binding site on integrin for the RGD sequence in fibronectin is located right in here. Okay. When this is erect, it can easily bind the integrin, the fibronectin sequences here. When it's curled over and the fibronectin sequences are up here, you can see there's not much chance of this actually recognizing the RGD amino acid sequence here in the fibronectin. Okay. Here, in more detail, is again fibronectin, one of the two identical chains of fibronectin. These two repeat sequences here, shown here, these are what we call, these are examples of what we call the immunoglobulin fold. It's one beta sheet structure on top of a second beta sheet structure. The beta sheets are flat. This is kind of like a sandwich. The bread is one beta sheet. The second piece of bread is a second beta sheet. And the thing that holds the two beta sheets sticking to each other is hydrophobic side chains from the amino acid sequences here and here. They interact with each other. So the hydrophobic side chains are like the butter. It's like a bread and butter sandwich, basically. It's a very stable repeat, and there are disulfide bonds that pass between the two pieces of bread. So here is one of these immunoglobulin folds. Here's a second one. There's a little loop here, and this little loop right here, we have this amino acid sequence, RGD. And this is the synergy piece. So when the integrin binds, we're going to bind the RGD sequence, and we're going to recognize this so-called synergy sequence on the adjacent little big box here. Okay. Now, going back to this. Cells can go back and forth between a low affinity to a high affinity state. When you go from low affinity to high affinity, that's called inside-out signaling. I'm going to show you in more detail. So going in this direction is inside-out signaling. Inside-out signaling, going this way. And going this way is so-called outside-in signaling. That's because internal signals can convert low affinity integrin to high affinity integrin. When high affinity integrin interacts with its ligands, it recruits signal transduction molecules down here on its tails that then initiate signals to the interior of the cell. So this is a signal coming from the outside world binding here, causing these guys to change their shape a little bit and initiate signal transduction events. So let's take a look at more detail of what I mean by this. Okay, so here is integrin in its low affinity state. And an internal signal generated typically by maybe some kind of cytokine or hormone of some sort here binding to one another. We initiate signaling events here, and then an internal event causes the integrins to go from their low affinity curled over state to their erect prime state. Okay, and then they cluster, and now we go from essentially low affinity to high affinity here. Then we interact with the extracellular matrix. Okay, that leads to further clustering, and if you get sufficient numbers of these clustered, you then recruit signal transduction molecules like the SARC tyrosine kinase, the fat tyrosine kinase, and then many other molecules here. So this here, the binding of the outside world extracellular matrix to the integrins initiates an outside in signaling event here. So we have inside out signaling and outside in signaling. And the only reason this could be a little bit confusing for many people is because the inside out signaling event is actually initiated by previous outside in signaling here, unrelated to the integrins, but related to cytokines and cytokines. I'm not going to talk about this in my immunology class, which I teach in the spring. We talk about this inside-out, outside-in signaling in the context of lymphocytes entering the lymph node, or in the context of neutrophils entering the site of an infection. We have an integrin called LFA1, a cytokine receptor called the CXCL8 receptor, and so forth. And in this case, when the integrin binds to its ligand, so we get the integrin from low affinity from the high affinity integrin, the high affinity integrin then can bind to its ligand, and then initially outside in signaling, and the outside in signaling events here, signaling 2 here, turns on genes and protein kinases and so forth that initiate the behavior of the cell called diapedesis, where it will now sort of melt its way through the adhesion molecules that join 2 blood vessel cells here and pass through the bloodstream, through the blood vessel cells, wall, the vascular endothelial cells, and come into their target destination. Okay. I'm not saying any more about this. The next page is just a repeat of what we were just saying. We also see these contractile structures of actin and myosin during cleavage of the 2 daughter cells and the swaying of the apical surface of the intestinal epithelium. So that then takes us out of back to our outline. We now talked in the context of broad context of non-muscle actin. We talked about tightly bundled actin and loosely bundled actin. Now we're going to talk about actin networks. Here, just very briefly here, without going into too much detail. If you sort of take a look, if you can make a grazing cut through the surface, in this case of a platelet, you can see underneath the plasma, so here's the plasma membrane, here's what's just beneath the plasma membrane. This stuff is a protein network called the spectrin network here. Underneath the spectrin network, which is a very springy meshwork of proteins, we have the actin cortex here, which is these orthogonal arrays of actin filaments. Here you look at a lamellipodia in more detail, you see these branched actin filaments, which I refer to. So in the lamellipodia here, we have these branched actin filaments. We'll talk more about that. The branch production involves proteins with names like ARC23 and FLOS, which we'll get to, I hope, even this morning. So here is an actin gel, or an actin network structure. Okay. It involves, of course, the actin filaments and their random orientation with respect to each other. And at certain points, one actin filament going from north and south will cross over another actin filament going, east to west here. Okay. And at these intersection nodes, actually the formation of the node and the stabilization of that node structure is due to a protein, kind of a cross-linking protein that we call filament. You should know filament for the test. Filament is shaped, has two globular actin binding heads and then two tails which are joined to each other, and that tail joint is very flexible. It can form an acute angle or an obtuse angle and so forth, adjusting itself to the orientation of the actin filaments. But wherever we have these little crossover networks, it's all stapled in place by filament. Now, whenever you have long filamentous polymers, it could be DNA, it can be RNA, it can be long filamentous proteins. If you have those macromolecules in aqueous solution, they form a very viscous solution. You can think of this as being, if you were a little Maxwell Demon-sized person, trying to cut your way through the jungle of vines and so forth. It's hard to push your way through all that stuff. Well, that's what you have when you have viscous, you measure viscosity, in fact, by having a long graduated cylinder filled with the viscous solution. You drop a little ball bearing in. If it's water, it falls like a bullet to the bottom of the graduated cylinder and cracks the bottom of the cylinder. If it's a viscous solution of DNA or RNA, it slowly makes its way through. DNA is almost like nose snot. It's very viscous. Now, there's a difference between a viscous solution versus what physicians call a gel. Certain viscous solutions can be so viscous that they are almost gel-like. But we make a distinction between a viscous solution and a gel, and the distinction is the cross-linking of the filaments. In the laboratory, you have made polyacrylamide gels. You add the subunit proteins together and the acrylamide monomers. You catalyze polymerization. There's some cross-linking component in that mixture as well. If graduate student TA left out the cross-linking component of that mixture, you'll get a viscous solution of acrylamide, but it won't be a gel. You need the cross-linking to form a gel. And this is basically, for example, if I'm a ball bearing, I'm huge compared to this structure here. If these are free to move, I could just push my way through them. But if I'm a big ball bearing and I get into this, I'm just going to bounce off this surface here because I can't push the filament molecules aside. They're anchored to each other. And that's what makes it a gel. So I'll be clear on the distinction between a gel versus a viscous solution. A gel, the filaments, the polymers, are cross-linked. And the cross-linking agent in the cell to form an actin gel is filament. Now, cells can convert the gel state of actin filaments in their cytoplasm. They can go back and forth between a gel state and a sol state. And for example, when an amoeba is moving through the solutions and sending out pseudopods, the cytoplasm of the amoeba is in a cross-linked gel state, and then locally it can go into a viscous solution state. And so when it goes from a gel to a sol, that viscous sol can sort of leap forward to form the pseudopod. And then you can cross-link the pseudopod back to the gel state, that kind of thing. Here, what's happening when you go back and forth, let's start from the gel state. We have all the active filaments cross-linked by filament. Now, in this mesh of actin filaments, this cross-linked mesh, we have a whole lot of these enzymes, these proteins called gel solid. As the name would imply, gel solid can convert a gel to a viscous solution. Which from gel to solution. Gel solid catalyzes the conversion of a gel state of actin filaments to a solution state, a viscous solution state. And as I told you about 1 billion times, the cytoplasm of all cells is basically calcium-free, the cytosol, calcium-free. And if we open calcium channels, when calcium appears in the cytosol, it acts, in fact, like a second messenger, like cyclic AMP or any other second messenger. So there are a number of enzymes and processes which are activated by the appearance of calcium. So when the amoeba senses its bacterial prey, little bacterial body parts are diffusing through, and they'll bind to receptors on the amoeba surface, and those receptors will open up calcium channels, and calcium will come flooding locally into the cytosol of that amoeba, and the calcium will then bind to gel solid. So here's now gel solid with a calcium ion in it. There's another calcium ion. So all these gel solids bind calcium and then become activated, and what they can then do is, when they have calcium bind to them here, they can come over here, cut the actin filament, okay, and then cap the newly created synapse. So here we have gel solids which have severed actin filaments, and then having severed the filament, there'll be a minus end and a plus end. If you sever the actin filament, they jump onto the plus end and they cap it, preventing any further polymerization of the actin filament. So calcium gel solid severs actin filaments and then caps the created plus ends. And you don't need to pay attention to any of the details on this picture. I just showed it to you briefly to give you the idea of an amoeba, because this is actually the original picture. He's imagining how this process might occur in the immune system cell, like a neutrophil, or maybe a macrophage. Okay, now, we talked about item number 1, we talked about item number 2. Item number 3, actin filaments are linked to membrane proteins. I'm going to go through this extremely briefly. Okay. There's a very famous interaction between actin filaments and the plasma membrane that's mediated by a protein called dystrophin. So here's the amino-terminal end of dystrophin, and up here is the carboxy-terminal end of dystrophin. And it binds actin filaments, and it binds certain extracellular adhesion matrix, adhesion molecules here in the muscle cell membrane. The genetic disease, muscular dystrophy, we finally track down what gene is mutated to produce this muscular dystrophy disease, and that gene encodes this protein, which was therefore called dystrophin, the dystrophin protein. And it's a very springy protein, so that when the muscles contract, you maintain a springy connection that's still to the plasma membrane. When you don't have this, you tend to punch holes in the plasma membrane when the muscles are contracting. We're going to then pass right over all the remaining several pages here, and then come to another set of lectures here. So now we're moving on to lecture notes that are a different Xerox set of them. We've talked then about item 1, item 2, and in the briefest way, item number 3, and then there's a whole section of notes here on the in vitro polymerization kinetics of actin. All very interesting and informative. In the interest of getting on with things, I'm going to just talk in the briefest way about this. Okay. have actin monomers and they're in solution, we can initiate polymerization by adding some ATP, and then actin monomers will, if they come in collide with each other at just the right direction, just the right speed, they can form a little trimer here. Now, having formed this is a pretty stable structure, but if a fast-moving actin monomer collides with that, it'll blow it all apart anyway. But once you start getting a trimer, it's a little bit more stable, and you can then get other monomers to add on and begin to form an actin filament here. And we have a rapidly growing end and a slowly growing end, as you know. And the actin filament will get longer with time. So you have to have a certain concentration of monomers to be able to initiate this polymerase. If this is too dilute, it's too improbable that you'll form this critical trimer structure. So there's an actin concentration dependent thing here. So we talk about the critical concentration of actin, above which we'll initiate polymerization, below which we can wait till the cows come home and we'll get the polymerization. Now, if we have stuff that's at the critical concentration or above the critical concentration, if we take a little bit of pieces of actin filament, okay, these would act as nucleating seeds, so to speak, add that to the test tube, these things will then explosively add on to the end of an existing short piece of actin. So if we don't have any nuclei, if we're above the critical concentration, no nuclei have been added, we'll start to polymerize just because we're above the critical concentration. But if we add little nuclei seeds here, we get explosive polymerization of the actin filaments. Okay, now then, in looking at the in vitro kinetics of actin polymerization, it was discovered that while we speak of a critical concentration below which we won't get actin polymerization, above which we will, we didn't fully appreciate for a number of years that actually the plus end requires less actin to be able to polymerize as compared to the minus end. Okay? So the critical concentration for polymerization at the plus end is to the left here, a low value on this concentration gradient here, whereas for the minus end, we require a higher concentration of monomer. So the fact that we have two different critical concentrations, C sub C, for the plus and minus ends of an actin filament, that can be demonstrated, and in previous years I have shown pictures like this annotated, and you've had to explain what's going on here. But I'm not going to do that for you on this upcoming exam 3. Okay? But this is all about showing that we have critical concentrations. When we're above the critical concentration for both the plus and minus ends, we get growth of the filament at both ends. If we're above the critical concentration for the plus end, but still below the concentration for the minus end, then we could get growth at the plus end, but we get falling apart at the minus end. That is some previous development. And this whole thing leads to a phenomenon which was discovered in the test tube, but we now know happens inside of cells. That is an important aspect of actin filament dynamics inside of cells, something called treadmilling. So in actin filament, there are conditions of actin inside the cell or inside the test tube where the plus end is growing, but the minus end is falling apart. So we're adding on to the plus end, but falling apart at the minus end. And in fact, there's one particular concentration of actin monomer where we have exactly balanced. The rate at which we're growing on the plus end is exactly counterbalanced by the rate at which we're falling apart at the minus end. So this is treadmilling in the specific case where the filament does not change its length over time, because these two on and off processes are exactly balanced. But treadmilling, we now know, goes on inside all the cells. It's not usually the case where we have exactly balanced plus and minus. But typically one end is growing and the other end is falling apart. Very often inside of cells. So I just want you to know that broad understanding of what we mean by treadmilling. I'm not going to give you my standard question where you have to sort of explain a diagram like this. Now I do want you to know about two drugs which affect actin polymerization kinetics inside of cells. One drug, a number of drugs, but one famous drug which inhibits actin filament polymerization and in fact induces depolymerization, okay, is a drug called cytokilasin D. You should know that for the test. Another drug has the opposite effect. It hyperstabilizes actin filaments and drives excessive polymerization. And this is a drug called phalloidin. I think both of these drugs come from fungi. In the particular case of the fungus from which we get this drug is, if you take some, it kill your liver pretty quickly. Okay? But it's a very nasty toxin. And the mushroom from which it is derived is called the angel of death mushroom, otherwise known as the death cap mushroom. So, graduate student, I had a postdoc friend who was an amateur mushroom collector and enjoyed finding all kinds of interesting mushrooms. And one night I took my life in my hands and had a mushroom. Dinner with him and his wife. Thank God he knew his mushrooms. I would never do that now. That's crazy. What was I thinking? Okay. Now then, that's all we want to say about actin polymerization and depolymerization in the test tube. But now we want to talk about what goes on inside of living cells, the regulation of actin polymerization in vivo. Okay. Now we're going to begin by talking about a pair of proteins that bind monomeric actin. And now we're on page number, just like page 132 in your notes. So here's actin. Here's a little cleft where the ATP sits in right here. Now, maybe I'll just begin by saying that inside of cells, there's such a high concentration of actin that you would think almost all the actin would be in the polymerized form, but that's not true. Only about half of the actin is polymerized as actin filaments. The rest of it is as a pool of monomeric actin. So obviously we're regulators, not just physical chemistry, as we see in the test tube. And there are a pair of proteins that we've come to know about that govern actin polymerization. One of them promotes actin filament polymerization. It's called profilin. There's profilaments, okay? It promotes actin filament formation. The other one is called thymosin. Now, it's actually called thymosin beta 4, but we're just going to call it thymosin for our purposes for the test and for my discussing. Well, see here, if this is an actin filament, we have a plus end of an actin filament and a minus end, and this is the orientation. Here's the ATP binding pocket in here, and we're going to add on a subunit that's going to come on here, and this little upper end here where the ATP is on would bind to the butt end of the actin monomer that's already become part of the filament. Thymosin binds right here. It kind of covers up that little binding pocket here, and you can see how it would prevent the interaction between the tips of this monomer here and the base of the existing monomer, which is part of the filament. You see that. So thymosin inhibits polymerization, and profilin promotes polymerization. And you'll need to remember that. Now, how does profilin promote polymerization? Pause while I have a drink. Is this little thing in your notes? I think maybe not. No? All you need to do is remember that thymosin, this little guy right here, T-H-Y-M, thymosin. Here's thymosin. Thymosin is covering up the actin monomer and preventing it from attaching to a non-filament. Now, inside the cell, we now know that the way thymosin promotes polymerization is by interacting with partner proteins inside the cells. And here's kind of the ongoing cycle. Here's an actin filament. Here's the plus end. We still have ATP in that binding pocket. But after an actin monomer becomes part of the filament, a small conformational change occurs in the actin monomer, and it slowly activates its ATPase activity, and it converts the ATP to ADP. So at the very tip of a growing actin filament, everything is in the ATP form. Deeper in, and all the way to the minus end, all the actin subunits are in the ADP form. And they have a lower affinity for each other. They have a different shape. These are very sticky to each other. These are less sticky to each other. So we have an off rate here, and we will release ADP actin here. Now, profilin recognizes ADP actin and binds to it. Here we go. It then catalyzes the release of the ADP nucleotide and the uptake of an ATP nucleotide. So now profilin catalyzes. This is sort of poorly, it doesn't like to grow. It's poorly able to add on to a minus end, it's not very sticky, and it's not very good at adding on to a plus end either. So this is pretty much kind of inert ADP actin subunit here. But profilin is converting it to, so to speak, active actin with ATP in its binding pocket. Now, this profilin actin complex can then come up here and interact with partner proteins that have names like WASP and formin. WASP works in conjunction with actin with proteins that look like actin. They're called actin-related proteins, ARPs. Structurally and amino acid sequences they're very similar to actin, but they're different. But they can interact with actin and promote the addition of actin onto a growing filament. And we'll talk about formin as well. So we can just keep this circle going on here. We can be releasing actin from one end, and then profilin comes into play. So profilin will then add it on here, and then the process be released. But we also have inside the cells this inhibitor, thymosin. And thymosin and profilin have about the same affinity for actin. This kind of depends on their relative concentration. So in a cell at a certain time, we might have much more thymosin than profilin. Or locally, we might have more thymosin than profilin. If we have an excess of thymosin will come up with a kid. The school guard at lunchtime and say, I like your lunch, and just takes it, okay? So thymosin kicks off profilin and grabs the ATP actin monomer and sequesters it. If at a later time profilin levels rise, okay, the profilin will come along and say, I'm going to take my lunch back. Okay. He displaces thymosin and he takes the actin molecule. There's a dynamic interaction. So, we're going to talk about some of these partners for profilin. We're going to start talking then about formins, as I recall. Yes, we'll talk first about formin. And then we'll talk, there's a family of these blocked by proteins that we're going to want to talk about. Okay. This is formin. This is the formin protein here. When it's curled up on itself like this, it is inactive. Now, formin can be activated by signal transduction events, and in particular, signal transduction events, not all of them, many signal transduction events can lead to the activation of a small G protein called Rho. Again, as I said on several occasions, you learned from Dr. Cont about the first one of these proteins that we discovered some 20 years ago, called RAS, R-A-S. Okay. RAS is turned on by growth factor receptors. Okay. And RAS then initiates the so-called famous MAP kinase cascade, turning on genes for entry to the cell cycle. After we discovered RAS, a sort of low homology, low relaxed stringency hybridization experiments, we discovered a family of genes that encode proteins that resemble RAS. Okay, and one of these is called Rho, and it's sort of the hallmark protein of a class of small G proteins called the Rho family of G proteins. And here's the premier guy himself. In domestic circumstances, he has GDP in his mouth, but signal transduction events cause him to spit out the GDP and take up GTP. Now when he has GTP in his mouth, he has the right shape to interact with a downstream target. And one of his downstream targets is formin. Formin has a Rho binding domain, RBD. And when active Rho binds formin, that causes formin to uncurl, okay, and expose its two so-called formin homology domains as FH1 and FH2. Now these two guys, in the structure of the formin protein as a whole, the FH1 and FH2 domains work as partners like this. They're like, each domain represents like half a donut, and then the other half a donut, and these two half donuts are linked flexibly to each other, and they can go up and down with respect to each other, like a little reciprocating piston action. So in this conformation, we could nestle an ATP actin right underneath that half donut, okay? And then one right underneath this half donut. Okay? And now we have a pocket for a third actin monomer to come in. Okay? When that happens, then we'll get the reciprocating action. This one with the third actin will drop down. This one will come up. Okay, so this used to be down. This used to be up, lift up and down. Now we have a pocket for another actin monomer. All the way up, and then we grow the actin filament with the formin protein associated with the rapidly polymerizing plus end, and greatly catalyzing the polymerization of actin monomers. Okay. Now we find formin activation of actin filaments in a number of locations. Very importantly, I pointed out to you, remember these focal adhesion structures, which are centers for growth factor signal production and so forth? I say when you cluster the integrin adhesion molecules, you recruit also these formin molecules here. So formins associate with the actin filament, and then they cause polymerization of the actin filament, and you kind of, plus that's kind of anchored in place there with the actin structure, but the protein, the filament's getting longer and longer and longer as we add more and more subunits right there at the focal adhesion structure. Now these actin filaments that grow out from the focal adhesion structures are these, because we've also recruited alpha-actinin, they're widely spaced and they can interact with myosin bipolar bundles. So formins catalyze the formation of these stress fibers, which are, remember, impactful structures. I had here, for those of you who are a little bit getting on your small G protein memories, here's the sort of story on Rho. The same thing we could be saying about RAS, okay? Small G proteins are activated by a GTP exchange factor, a GEF. Now that's the broad name for them, okay? For RAS, the GEF can be, remember, it's called the SOS protein. Doesn't matter. So a GEF will cause a small G protein to spit out its GDP and then take up GTP and then close its mouth. And now it's in a different shape than it was before. In this new active state, it has a different shape. Now, it doesn't, many people think that small G proteins will phosphorylate their targets because everybody's always phosphorylating something. G Rhos do not phosphorylate something, they simply stick to certain downstream partner proteins. And they stick because the little bubble, the nipple is popped out here, in its active state, and it can fit into a slot on very specific target proteins. And for example, formin could be one of the target proteins that this row could bind to, and merely by binding to it, activate it. Now we also have proteins which facilitate the intrinsic GTPase activity here. These are called GTPase activating proteins, GAPs as they're all down here. So GAP proteins will then convert the active G protein to inactive G protein. And we also have some subproteins which will sequester the inactive G protein. Guanine nucleotide displacement inhibitors, GDIs here, which will remove this row from the scene altogether. So that was just so that you remember all this stuff. Now then, I'm making pretty progress here. I love this. Here, so we talked about what's happening inside of cells, regulating active filament concentration. And I said that includes WASP family proteins and formins. We talked about formins. Now I'm going to talk about WASP, really, these WASP proteins. And there are two important family members here. WASP itself. WASP takes its name from a genetic disease involving poor immune system, the Wiskott-Aldrich syndrome protein. So in the children born with this disease, their lymphocytes are unable to migrate properly, just like the infection that is getting their lymphocytes properly. They have a defect in their migration pathways. So they track down the gene, and the gene encodes this protein, and here it is. Here's this protein, the WASP protein. Now we find variants of WASP in different cells. Okay, this is a form I think found in neurons, and that's why it's called, I think, NWASP. Well, we're not going to worry about whether it's NWASP or whatever. It's just WASP as far as we're concerned, which the term that is used disparagingly for white people on the East Coast, white Anglo-Saxon Protestants, Jewish members of our society 100 years ago would refer to those dead wasps. But this is not white Anglo-Saxon Protestant. This is Wiskott-Aldrich syndrome protein. Now then, of course, I'm almost a wasp, except I'm a white Anglo-Saxon Catholic, so but pretty close to a wasp. So we have Wiskott-Aldrich syndrome protein. Here it is, kind of like formin, curled up on itself, inactive. And it's going to become activated by another G protein. In this case, another member of the Rho family called CDC42. Here is inactive CDC42 in its GDP state. And here's active CDC42 with GTP in its mouth. And now it has the right shape to interact with the G protein binding domain here. Here it is, here's the G protein binding domain. That this domain will bind to activated CDC42 that will cause WASP to uncurl. Okay. Now it has a profilin binding domain. Okay. And it recruits active profilin complexes here, and then delivers them to a region right here where we bind so-called ARP2/3 complex here. The ARP2/3 complex, again, it has these two proteins that closely resemble the structure and everything else. Actin itself, but they're not actin, they're actin-related proteins, and there are two of them on coded by two different genes, ARP2 and then ARP3, and together they stick to each other, and they're held in just the right conformation with respect to each other, and their shape is activated by kind of scaffolding proteins, and we call the whole thing the ARP2/3 complex, which will also bind to WASP. And here we then recruit profilin actin complexes. We strip off the profilin and hide out the actin to the ARP2/3 complex, which will then initiate polymerization of the actin filament. Now, in most cells, we activate WASP via CDC42. In some cells, we use an alternate pathway where a protein called the WISH protein comes into play. Don't worry about WISH, not on the test. Scratch this out, not on the test. I just want you to remember WASP and CDC42. And I want you to remember that when we use the wasp scaffolding protein here, the filaments that we produce are going to be involved in these tightly packed actin bundles, okay? Remember filopodia and microvilli and so forth? Those actin filaments are catalyzed, their polymerization is catalyzed like wasp. Now then, there is another member of the wasp family called wave. These guys have so much fun when we discovered this. This one was called wave. Wave kind of looks like a wasp, quite different from yours, but it's curled up on itself, and it's going to be activated by the third member of the row family. So the row family are usually the three principal members are RAF, row, and CDC42. We'll come back to this in a moment. Typically, RAF will become activated first. RAF will in turn lead to the activation of row, and then row will turn on CDC42. They often work sequentially with each other. So here's RAF. Here it is inactive with GDP. There's a signal transduction event here. In this case, it will be a tyrosine kinase receptor. It doesn't always have to be necessarily this. And so that will activate a GEF, which will bind to RAC, make RAC spit out its GDP, and take up GTP. Now, GTP RAC. Now, in this case, the small G protein doesn't directly interact with the RAC, with the wave. There's a kind of adapter protein we call IRSP53. You don't need to remember that. I just want you to remember that you're going to remember that it is Rho that activates formin, that it is CDC42 that activates WASP, okay? And then it is RAC that activates the wave. So, RAC activates wave, okay? And the same deal here, we have the domains here that will bind the profilin actin complex here, strip the profilin off, deliver the actin to the ARP23 complex, and we will initiate actin filament polymerization. But the wave complex, the actin filaments whose formation it catalyzes are these branched actin filaments that we find in lamellipodia formation. And here's a picture that you're not going to have to memorize for the test, obviously. It's from a little review article now. At the time this review article came out, the guys didn't realize that the G protein involved is actually RAC here, and the family member here is not WASP but WAVE, but never mind all that. We'll have WAVE being activated by RAC, okay? We start to initiate the ARP23 complex. Here's the actin profilin complexes here. We're going to then permit the actin in their ATP forms to start to interact with the ARP23 proteins here. Okay? And as we do that, as soon as we start grabbing actin monomers, starting to form an actin filament, this complex here loses its affinity to the wave scaffold here, and will jump off and land onto the walls of a nearby actin filament. Okay, so here it is. It's jumped off and landed onto here. Okay, and now we continue the polymerization on the wall while we're attached at our base to the walls of the existing actin filament. So we have formed a branch here with a characteristic 70-degree angle. We do not have to remember 70-degree angle, but we formed a branch here. And we'll just keep doing that branching process. And that's how we extend the spin flat sheets of actin, branched actin sheets coated with plasma membrane. These are the lamella coated. Early cell biologists call these the ruffled membranes that they receive from cells. And then, as these branched actin sheets are forming underneath the plasma membrane and growing outwards, and their interior deeper into the cell, we now have actin in the ADP form here, and then there are molecules that will sort of things like ADF another complex here, cofilin, ADF and cofilin are the same thing, that will sever ADP actin filaments into bits and pieces here, and then cause the depolymerization. So we're at the interior base of this thin sheet of branched actin. Things are falling apart. We're releasing monomeric actin, which can then diffuse and then add on to the growing end. So it is a treadmill that's going on. We're growing at the plus end and we're falling apart at the minus end. And so here's a picture of your book that kind of summarizes what I've said about the movement of fibroblasts. We extend filopodia and lamellipodia forward. They have adhesion molecules at their tips, and they'll adhere to the extracellular matrix that's coating the petri dish. We form new focal adhesion structures here. The stress fibers polymerize out from that, and we're going to get contractile things here, and we're going to pull the butt end forward, and then continue moving forward. And as we go here, we're pulling on the rear end. We break down the focal adhesion structures at the back end of the cell, time-coordinated way somehow. Sometimes we can't disassemble the old focal adhesion structures fast enough, and we leave behind only the pieces of the cell that adhere to the dish, as I was saying. So I did pretty good. I didn't get quite finished, but so when I see you again on Wednesday, I'll finish up this last little bit on the things that we've been talking about. And